Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Oriana Tarova from Thermo Fisher Scientific.
Hi everyone, thanks for being here today. Just a little bit about me. I am the product manager for the Purification Chromatography Resins within the Bioproduction division at Thermo Fisher Scientific. I've been with Thermo for about 9 years. I am a chemist by training with technical background in small molecule and bead chemistries. Primarily my responsibilities now are to maintain and grow the Porous chromatography resin product line. In this webinar today, I'll be talking about the custom resin programs within Purification at Thermo Fisher Scientific, and I will then provide a specific example of how these solutions are utilized in gene therapy applications. So here at Thermo Fisher, we serve a broad range of customers, and if we were to look at our customers' goals, most of what they have in common is to increase innovation and productivity. We are proud to be able to combine our capabilities and fulfill our mission, which is to enable our customers to make the world healthier, cleaner, and safer. As far as our value proposition, our customers are at the center of everything that we do to help them accelerate innovation and enhance productivity. And the products and services are very important, but the actual benefits that they offer to our customers are what ultimately allows us to deliver on our commitment and our value proposition. So with that said, I would like to provide an overview of what we first offer in terms of our bioproduction division, and then the focus will be in purification. But as you see in this slide, at Thermo Fisher bioproduction division, we have industry-leading capabilities across the bioprocessing workflow from preclinical through commercial manufacturing. So upstream at cell line development and media optimization stage, we enable our customers to meet their process development faster by providing PD services and media optimization services. At mixing, cell culture, and fermentation stage, our GIPCO cell culture products and Sera products are well positioned to help increase titers. And also our quality single-use mixers and bioreactors and bags to increase productivity and flexibility. Of course, downstream, which will be the focus during this webinar, as far as purification of biomolecules, our porous chromatography resins and the Capture Select affinity ligands and resins offer high capacity, high specificity, and superior resolution to maximize downstream purification efficiency. So let's expand a little bit more on our purification capabilities. So our core capabilities are in ligand and resin development as well as manufacturing. By utilizing our purification solutions, customers have developed platform purification processes that have resulted in increased productivity and improved product yield for various applications. The Capture Select affinity ligands and resins are developed at our site in the Netherlands. We have established ligand manufacturing capabilities in Naarden in the Netherlands since 1997. Upstream vessels range from 10 liters to 15,000 liters. The porous chromatography resins, however, are developed and manufactured in Bedford, Massachusetts at the US site. We have successful track record in resin development, scale-up, and manufacturing for over 2 decades. We have a dedicated pilot plant facility for the custom resin programs, which I will further describe in my upcoming slides. And also we have the capability to scale up resin manufacturing up to 250-liter commercial lot sizes. So now that I have provided a high-level overview of our purification tools and capabilities, I would like to further focus on how we actually provide value downstream. And as we talk about delivering value downstream, I'd like to start by looking at some of the industry trends that are driving growth in downstream purification. Certainly, mAb-based therapeutics continue to dominate, even with new cell lines emerging. So there is more mAb fragments, Fabs, ADCs. As far as vaccine production, processes are being redesigned to be more responsive, more reproducible. There is interest in modernizing legacy processes in plasma. There is growth in biosimilars as more drugs are coming off patent. Of course, growing interest in the area of gene therapy. The market is expected to grow at about 15% CAGR. Speed to clinic is the main product development driver, and quality and efficiency will always remain critical. The focus today will be on the gene therapy applications. So like I said in the previous slide, there has been an increased interest and focus in gene therapy applications over the past decade. Gene therapies promise versatile treatment of long-term and potentially curative benefits to patients with genetic or acquired diseases. Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. And several types of viruses can be used as delivery vehicles to infect and transfer a functional gene into patient cells. However, viral vectors and the adeno-associated virus, so the AAVs, have emerged as the vector of choice for many therapies because it can mediate primarily long-term tissue-specific gene expression with low immunogenicity. Of course, when we talk about this exciting application, efficient commercial manufacturing capabilities for these unique therapies to increase productivity and ultimately to meet market needs is very critical. And one major challenge that the field faces is the lack of industrialized platform technologies to purify viral vectors. Although the biopharmaceutical industry is primed to purify viral vectors on a scale suitable for clinical supply or orphan indications, there is still a lot of work that is being done towards generating industrialized platform technologies that will maximize productivity and enable efficient, simple, and inexpensive purification of biologically active viral vectors for large-scale commercial manufacturing. So with that said, the current purification methods for viral vector manufacturing consists of multiple steps that can include several chromatography steps like I'm showing here in this slide. For example, ion exchange chromatography or hydrophobic interaction, IMAC, Affinity, heparin, those are all options that have been used. Such approach, however, will lead to long process development lead times, a more expensive process, and also, which is very critical, cumulative yield losses. Cesium chloride density gradient centrifugation is also used as an alternative. Iodexanol gradients are also utilized in viral vector purification. These processes, however, suffer from poor scalability, and this is limiting the commercial feasibility of any processing viral vector. So in order to address the challenges that I mentioned in the previous slide, Thermo Fisher Scientific offers the Capture Select technology, which is used to produce high binding ligands for affinity chromatography. So this form of chromatography is proving to be essential purification platform for many biotherapeutic molecules because it enables a high purity and high productivity process with fewer purification steps than traditional processes, while still offering process consistency and scalability. I think we're all familiar with the affinity chromatography principle, so I do not need to go into a lot of detail with that one. But what is important here to point out is that the Capture Select products offer antibody-based target specificity, and the performance is independent of feedstock. For example, if we look at the factor VIII example that I've highlighted here in this slide, similar selectivity is shown in load feedstock cell line, which is the left side and also in plasma, which is to the right. So with that said, I spoke about the Capture Select Affinity solutions, and we offer a full portfolio of products for biopharmaceutical development from discovery through to screening and final manufacturing. But I would like to zoom in a little bit more and further describe the technology. So the Capture Select technology is based on the variable domain of camelid heavy chain only antibodies, so the VHH, which as single antibody domains, they provide full functionality in antigen-specific recognition and high affinity binding. Also, due to their compact structure, the domains are very robust and can withstand the various conditions as are typically run in chromatography. During Ligand Discovery, which I will explain our program a little bit more in detail in the upcoming slides, but the final ligand that meets all predefined requirements is selected and produced in an animal origin-free system in baker's yeast. And we have a proven Ligand Discovery program to develop the custom affinity solutions as well. And in addition to that, I would like to point out that our products are in commercial purification processes both in the US and in Europe. So putting it all together with what I've just described, we enable a paradigm shift in viral vector purification when using the Capture Select technology. The standard purification process, as I mentioned earlier, requires multiple chromatographic, filtration, and capture steps, which results in low yield and long process development timeline. When utilizing the Capture Select technology, high purity in a single step is obtained because your process is simplified, as I'm showing here in this slide. It shortens the development timelines and increases speed of therapeutics to the market. So essentially, from multiple chromatographic steps, we can do affinity chromatography, one capture step, and some customers will go straight to concentration and formulation. Other customers may do a polish step right after affinity chromatography. But overall, the feedback that we have received is that product yields have been increased from 20 to 60%, and cost has been reduced by a factor of 6 over alternative methods when utilizing our Porous Capture Select resins for viral vector purification. This result is crucial because the industry is focusing on developing industrial capabilities to produce these viral vectors in large amounts to meet clinical and future market demand. So now I'll expand a little bit more on the Affinity Ligand Development Program. And what I'd like to quickly go through here is really the structured approach that we follow when we develop these affinity ligands. During the first stage, which we refer to as work package 1, consists on the discovery of the VHH binding fragments, which occurs by immunization and library construction. This is then followed by hit screening, starting with a new immune library or an existing one. And then in 4 weeks, we'll have single clones binding to the protein of interest, showing the desired specificity. A successful hit screening is almost a guarantee for a positive outcome of the program. We then move to work package 2, which consists of library screening where phase selectivity, elution conditions, and stability are evaluated. This is a critical stage as various critical parameters are evaluated. And the lead screening is conducted by a unique 96 well plate array. The kinetic data of 96 candidates is analyzed for on and off rates. As I'm showing here in this slide, just a quick picture of what that looks like. The technique utilizes surface plasmon resonance, which is a labor-free detection method where molecular interactions may be analyzed on the surface. During this step, parameters such as specificity, binding affinity, elution conditions, and stability are evaluated. Again, this is a critical stage because the selection of ligand candidates for prototype resin testing in the next step is based on. So with that said, then the next step would be work package 3. And in this stage, 6 prototype ligands are cloned into our yeast expression system and produced and evaluated at Thermo Fisher Scientific in small-scale affinity chromatography. From there, we move on to the next stage, work package 4, and based on the results and discussions with our customers, 2 ligands are picked and produced at larger scale. Resin is prepared for evaluation at the customer site as well. Following work package 4, the customer will have access to an R&D scale resin that we can produce up to liter scale quantities. And then finally, the last stage is work package 5, where we work towards making the resin suitable for clinical and commercial manufacturing, of course. This includes things like setting up a robust manufacturing process for the ligand, developing prototype resins, development of product-specific leakage ELISAs. We do validation. We compile all the necessary data for the ligand and the resin to also generate a regulatory support file. Once we have the ligand, then we would immobilize it on a porous backbone, which is polystyrene divinyl benzene backbone. And so I would like to just quickly describe the porous technology, and then I'll further describe specifically the 2 affinity resins for viral vector purification. But just a little bit about porous technology. I would like to focus on 3 key attributes that differentiate porous from other chromatography resins. The first one is that it's made of polystyrene divinyl benzene, so the beads are rigid and incompressible. This is important because it makes them easy to handle and to pack and to maintain the packed bed height. Also, due to the polymeric nature of the backbone and the covalent chemistries that we attach to the backbone, the beads have very robust physical and chemical stability, and so they can be exposed to various chromatographic conditions and aggressive solutions that are used for cleaning, for cleanability. The second important attribute that I'd like to point out is the fact that they have a large pore structure, which unlocks the bead interior, increasing the surface area, which then results in higher capacity. Also, due to the large through-pore structures, the beads have improved mass transfer capability, allowing for ultimately a more efficient bead. So we can maintain high capacity, high resolution, independent of the operating flow rate, and this leads to efficiency, increased linear flow rates, and improves the productivity for our customers. And over here, I'm just showing an example of porous excess that has superior capacity to the other commercially available resins. And then lastly, the last differentiating feature of porous is that the average particle size is 50 micron. So it's a smaller particle size, but it allows for less band broadening in packed beds, and so therefore the ability to separate proteins more efficiently. The 50 micron particle size delivers unprecedented resolution, which is independent of scale as compared to other traditional resins. And we are comparing here porous excess, let's say, versus SP Sapphiros fast flow, where baseline resolution is maintained with porous. So again, putting it all together, what I'm showing here is that the porous and the capture select technologies are successfully combined to develop 2 affinity resins specifically engineered for the purification of adeno-associated virus subclasses 8 and 9. The AAV8 and AAV9 capture select ligands are immobilized on the high-performing porous R250 base bead. These resins outperform any product in the market for the purification of AAV8 and AAV9 viral vectors with respect to specificity and capacity. I'm showing here in a summary in the table of the capacities that our resins offer. We have performed capacity studies with a collaborator as well, and in addition we've received customer feedback from various evaluations that indeed these capacities can be obtained and even higher in some cases. So when utilizing these two resins, one step, AAV8 and AAV9 purification from crude material with high purity and yield will be obtained, and then that is frequently followed by a concentration step or polish step depending on the application. So let's expand a little bit more on the performance attributes for Porous Capture Select AAV8 and AAV9 resins, things such as purity and vector recovery. What we are showing here is some data that we generated in a collaborative study with Genethon. And this is looking at the purification of an AAV9 viral vector. And to the left, we are showing purity data where we compared multiple ion exchange steps with Porous Capture Select AAV9. So again, equivalent purity was obtained using both methods. However, the other graph shows that higher vector recovery is obtained when using Porous Capture Select AAV9. So for example, here we are showing a satisfactory vector recovery of greater than 80% that it was obtained. And also it's important to point out that no residual proteins were detected. So as I mentioned during the technology slide when I was describing the Porous technology, Porous is fully scalable with excellent pressure flow properties. The performance is independent of both the scale and the flow rate of the operation. Pressure flow curves of Porous are linear and predictable. Again, as I mentioned before, the beads are incompressible, so we can maintain very high operating linear flow rates with a modest pressure drop. And as we're showing here in the slide, you can operate Porous Capture Select AAV8 and AAV9 at fast flow rates with no back pressure issues. This was a 22 centimeter length column which generated less than 3 bar of pressure at 700 centimeters per hour. Customers have been able to scale up with no issue up to 200 liter scale. Porous Capture Select AAV8 and AAV9 Affinity resins also offer excellent resin lot-to-lot reproducibility, yielding in reproducible chromatography. Again, in this slide, we are showing some of these key points through some data that we generated with Genethon. And vector recovery data, and again this example is with AAV9. The data was collected at 3 different scales, generate 10 liter, 50 liter, and 200 liter to assess reproducibility of the process and to show scalability. And again, satisfactory vector recovery was obtained of greater than 70%. So to summarize quickly on the affinity piece, so Porous Capture Select AAV Affinity resins, these purification solutions will enable the design of platform processes and large-scale production processes for vector purification to increase productivity in order to meet market needs. I think this will help overcome the challenge that the field is currently facing as far as establishing commercial manufacturing capabilities for these unique therapies. And you know, as I mentioned earlier, affinity chromatography for purification of these biomolecules offers clear advantages that I hope I have shown those in my previous slide of obtaining high purity, high yield in a single step. We strongly believe that utilizing Porous Capture Select AAV8 and AAV9 affinity resins will be a significant improvement to the downstream processing by reducing purification steps and maximizing productivity. So now I would like to spend just a few minutes and talk about the next step after affinity chromatography. So I mentioned earlier typically the downstream purification for AAVs would be affinity chromatography after clarification, and then frequently is followed by concentration and sterile filtration. But at times a Polish chromatography step is conducted after the capture step. And this is primarily related to another challenge during AAV production, which has to do with the fact that empty capsids typically co-purify with genome-containing AAV vectors, which would interfere with transduction. Different methods have been reported to remove empty capsids from genome-containing vector particles by anion exchange chromatography. And there has also been shown that the Porous Q anion exchange resins can be used for these applications. All anion exchange resins bind and elute the desired AAV capsid and can be used as a polishing step after capture. So in this paper that I am referencing here in this slide, Porous HQ, for example, was optimized to efficiently separate empty capsids. It was observed that the early fractions, which were the less ionic fractions, contained empty capsids, and the genome-containing vector particles were in later fractions, for example. So I would like to conclude by really summarizing some of the points that I tried to make in this presentation. So the combination of our Porous technology and the Capture Select technology provides enabling purification tools to solve the manufacturing challenges in viral vector purification. Additionally, we offer an industrialized platform for AAV large-scale production to support market needs. The high specificity and capacity of Porous Capture Select AAV8 and AAV9 affinity resins offers the high purity in a single step from crude material. Additionally, our Porous Polish anion exchange resins offer high resolution and capacity, as well as salt tolerance that enable the robust separation of product from process and product-related impurities. So through Thermo Fisher's purification solutions, we want to improve process flow and reduce processing time, ultimately reducing cost of goods and enabling speed to market. Our resins, of course, are manufactured in state-of-the-art facility, and robust quality of our chromatography resins and processing is obtained on a daily basis. And additionally, we continue to provide the support and the technical engagement of our customers to help them solve any technical challenges that they face and ultimately help them obtain their productivity goals. So I would like to finish by thanking everybody for your time. This was the end of the webinar, and I think with that said, I think we can start taking questions.
Yes. So the first question, excuse me, the first question is, what are the advantages of Porous Capture Select resins compared to other resins on the market?
Sure. So I think it goes back to some of the key points that I was highlighting earlier. So first, I would like to point out the selectivity that comes from our Capture Select technology. And then, of course, you combine that with Porous, which has the rigid backbone. Performance is maintained through a range of linear flow rates. The large through-pore structure that provides superior capacity, and also the 50 micron particle size provides resolution are really all key benefits that differentiate our resins in the market. Really, we're able to successfully combine the affinity technology, which is Capture Select, with our superior performance on the polish side, which is the porous resins, and ultimately provide the entire solution in downstream purification.
Are there other resins available in the gene therapy space?
There are. There is one other affinity resin that is offered by GE Healthcare, that is AVB Cephalose.
And does Thermo have any others?
So we are currently developing additional resins that will target, again, gene therapy and cell therapy applications. The next resin that we will launch next year is a resin that will target purification of other AAV serotypes. So right now, Porous Capture Select AAV8 and AAV9 are suitable for AAV8 and AAV9 viral vectors. Our next generation resin will target additional serotypes. That's one resin that is in development right now, and we plan to launch next year. In addition to that, we're also working on a lentivirus product as well to enable lentiviral purification additionally.
Okay, and we have a couple questions on that. One person has the Porous AAV9, and she says the capacity is shown as 10 times for the AAV9, and I don't, maybe over something else, and she wants confirmation. Do you see that question?
Yep. So actually, thank you for that question, because I believe I mentioned it a little bit earlier, but perhaps it wasn't clear. So we have determined our capacity information that we have published in our product user guides. So it's 10 to the 13 for AV8 and 10 to the 14 for AV9. And we've confirmed this with customers and collaborators. However, we also get continuous feedback that I don't think customers have achieved maximum capacity. So I believe when they're saying that capacity is shown high for AAV9, it is possible because we also get that feedback from customers that they tend to get higher capacity. Unless the question is that he's not getting as high of capacity.
I think the question is probably related to the data that you showed with the, that published data. So.
But we can contact if the email address is available. I would actually like to contact to obtain some more detail what kind of capacity they're getting, and then we can also connect with our field application specialists, because they can help if they're not getting as high capacity. We can provide information there as well.
Okay, perfect. It looks like we have that information.
Great.
Do you have any other resins recommended for lentivirus with VSVG?
Yes. So that is one that is currently in development. The timelines when that is coming out, I cannot discuss at this time, but that is definitely in our pipeline to enable lentiviral virus purification. So it is a product that we are developing right now.
Okay. And then as far as your distribution, can these resins be obtained globally?
Yes, absolutely. So the resins are manufactured in Bedford. The Capture Select ligand is manufactured in Leiden. In Naarden in the Netherlands. But we have global capabilities to distribute the resins in every part of the world. We have distribution centers in US, in Europe, in APAC, so it is a non-issue.
Okay. Very good. So thank you, Oriana, and thank you to the audience for joining us.
Great.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at our future BioProcess International Ask the Expert webcasts. We have a full lineup covering many aspects of bioprocessing scheduled into 2017. Look for those announcements in your inbox. Thanks.